Now we will discuss types of immunoprecipitation. There are mainly three types which we will discuss here. Co-immunoprecipitation, then is chromatin immunoprecipitation, and then in the end we will discuss ribonucleoprotein immunoprecipitation. So first of all, co-immunoprecipitation. This co-immunoprecipitation is isolation of a protein or antigen along with any other protein or ligand bound to it. So we separate whole of the complex instead of isolating one protein or one antigen at a time. So whole of the complex is isolated. So it is separation of the entire protein complex out of a cell or tissue homogenate using a specific antibody. And afterwards, the unknown members of the complexes, they are further identified and characterized. So we can see if there are 4 or 5 proteins, for example, protein A, protein B, protein C, protein D, and protein E, they interact with each other to form one complex. So what we do, we separate this whole complex from the homogenate, and then each of the component from A to E, that is identified and characterized. It is used to analyze protein-protein interactions by using target-specific antibodies, and problems arise by interaction of various components of co-immunoprecipitations. Since these different components, they interact with each other, sometimes it is difficult to identify these components due to their interactions. This co-immunoprecipitation is shown here. You can see on the left side, when the cells are lysed, tissues are lysed, we get a mixture of different antigens, proteins, which may be carbohydrate, which may be proteins, which may be nucleic acid, so on. So here we can see on the left side, there are 2 proteins, protein X, protein Y. So they interact with each other. And by using these protein A coated agarose beads coated with protein A or protein G, we isolate this complex of 2 proteins. I mean, this complex of protein X and complex of protein Y. So, चूंकि यहाँ antibodies जो होती हैं, which are shown in red color, Y-shaped red color, ये beads के ऊपर coated हैं, तो यहाँ जो respective antigen है, जो protein है, उसके साथ bind करेगी, और बाकी जो components होंगे वो bind नहीं करेंगे. जब हम इस homogenate को centrifuge करते हैं, तो जो beads हैं, along with this antibody antigen complex, वो sediment हो जाएगा, और जो बाकी इसमें जो components होंगे वो separate हो जाएंगे. So अब हम थर्ड फेज में देखते हैं कि यहाँ सिर्फ वो जो प्रोटीन हैं वो सेपरेट हो गई along with the agarose beads coated with protein A, और उसके साथ हम जो एंटीजेंस या प्रोटीन X और Y सेपरेट हुए देख रहे हैं. इनको हम फर्दर कैरेक्टराइज कर सकते हैं बाय सेपरेटिंग दिस प्रोटीन्स फ्रॉम द बीड्स. तो एंड पे दिखाया गया बीड सेपरेट हो गई और ये प्रोटीन्स हैं वो सेपरेट हो गई. अब इन प्रोटीन्स को हम फर्दर प्रेसिपिटेट करके इनको फर्दर कैरेक्टराइज कर सकते हैं थ्रू STS पेज, थ्रू वेस्टर्न ब्लॉटिंग. एंटीबॉडी binds with its target protein or antigen through a particular epitope, and each component of the protein complex is identified with the successive immunoprecipitations. तो जाहिर है जैसे जैसे हम उनको स्टेप्स करते हैं, सक्सेसिव इम्यूनोप्रेसिपिटेशन करते हैं, तो हर एक जो कंपोनेंट है उसको आइडेंटिफाई किया जा सकता है बाय यूजिंग रिस्पेक्टिव एंटीबॉडीज अगेंस्ट दोज एंटीजेंस. Co-immunoprecipitation is repeated for the valid identification of components of the complex. जाहिर है एंटीबॉडीज चूँकि स्पेसिफिक होती हैं अगेंस्ट देयर एंटीजेंस, तो उनको यूज़ करते हुए हम आइडेंटिफाई उनको कर सकते हैं. Problems with co-immunoprecipitation may be resolved by changing the ionic strength of the buffers. जाहिर है उनकी जो स्ट्रेंथ है वो कम करके ज़्यादा करके, या by decreasing concentration of the antibody, उसको भी हम कम ज़्यादा करके, and similarly by pre-clearing the cell or tissue homogenate, इस तरह से करके भी हम co-immunoprecipitation के जो प्रॉब्लम होते हैं उनको रिजॉल्व कर सकते हैं।